I am Donatella Sgubin and I'm going to discuss our chapter Brain Tumor Stem Cells by Francesco Di Meco, Alessandro Perin, Donatella Sgubin and Alfredo Quiñones Hinojosa. These are our key concepts. Glioblastoma is the most malignant and frequent primary brain tumor. Glioblastoma cells are hierarchically organized with brain tumor stem cells on top. These cells share stem properties with physiological neurostem cells and are tumorigenic when implanted into immunodepressed mouse models. It has been proposed that brain tumor stem cells may arise from the subventricular zone of the lateral ventricles, but a comprehensive marker to identify them is still unknown. Given the numerous similarities between normal stem cells and brain tumor stem cells, we can potentially exploit the bulk of knowledge about normal stem cell biology to identify any possible novel molecular targets that could arrest brain tumor stem cells proliferation, such as inhibitors of the sonic hedgehog pathway required for the proliferation of stem cells, induction of terminal brain tumor stem cells differentiation using bone morphogenic protein, and disruption of the critical angiogenic niche of the brain tumor stem cells. The adult brain was considered a static organ without any cell proliferation and differentiation in the postnatal period. Altman revised this dogma, identifying dividing cells in restricted areas of neonatal rat brains. Years later, Reynolds and Weiss introduced the neurosphere forming assay to isolate in vitro neurostem cells from fetal and adult brain tissues. The brain tissue is dissociated into a single cell suspension and cultured in serum-free media supplemented with mitogens such as EGF and FGF. Under these conditions, neurostem cells are able to divide symmetrically and asymmetrically, forming spheres called neurospheres. A neurosphere contains the parental neurostem cells and the rapidly proliferating and more differentiated progeny. Under specific pro-differentiation culture conditions, these cells are able to differentiate into neurons, oligodendrocytes, and astrocytes. The sphere-forming ability of one single cell is assessed to determine stemness. This assay allows for the identification and isolation of all physiological neurostem cells from neural cell in the subventricular zone of the lateral ventricle and the subgranular layer of the hippocampal dentate gyrus. In contrast to traditional beliefs in oncology, the concept that only a small fraction of neoplastic cells, tumor stem cells, are responsible for tumorigenesis was proposed in 1963 when first observed in lymphoma cells. This concept quickly spread to other types of malignancies. Some peculiar glioblastoma-derived cells isolated from the clonogenic assay have stem properties and act as tumor-initiating cells, establishing tumor that closely resemble the main histologic, cytologic, and architectural features of the human disease. A correlation between the sphere-forming ability in vitro and tumor aggressiveness in vivo had been demonstrated. Not all cells within brain tumors have the ability to initiate tumorigenesis. Rather, brain tumor cells are hierarchically organized, with stem cells at the top of the hierarchy and differentiated into the aberrant tumor stromal cells at the bottom. Strong evidence now suggests that brain tumor stem cells express a collection of markers that differ from the tumor stromal cell counterparts. Markers once considered reliable for selecting only adult neurostem cells are also now adequate to label brain tumor stem cells as well. Brain tumor stem cells and neurostem cells marker spectrum overlaps in terms of intracellular protein such as nestin, ODIC2, MYC, SOX2, and surface markers such as CD133, CD44, etc. A single comprehensive brain tumor stem cell marker remains to be identified. There are three possible explanations for the origin of brain tumor stem cells. The traditional clonal evolution hypothesis argues that brain tumors arise from the dedifferentiation of dedicated adult brain cells. The subsequent clonal expansion of this single dedifferentiated cell and the accumulation of mutation over the time confer the progeny with the ability to proliferate and regenerate in an unregulated manner. This results in the eventual formation of the brain tumor stem cells, which go on to produce the primary tumor focus and migrate locally to ensure metastatic disease. With the discovery of adult neurostem cells and the observation that brain tumor stem cells behave much like adult neurostem cells in way of organization, proliferation, and regeneration, it was hypothesized that abnormal differentiation of adult neurostem cells may be the direct source of human brain tumors. The progenitor recruitment hypothesis argues that a single tumor progenitor cell of an undefined origin has the ability to recruit normally quiescent neuroprogenitor cells and induce them to proliferate in an unregulated manner through the release of local growth factors. Definitive proofs has yet to be found as to which hypothesis is dominant in the natural pathogenesis of human brain tumors, and there is significant experimental evidence to support all three alternatives. There is considerable evidence to support the idea that dedifferentiation and accumulation of genetic mutations within terminally committed adult brain cells may be the source of human brain tumor stem cells. The striking phenotypic similarities between neurostem cells and brain tumor stem cells make it tempting to conclude that brain tumor stem cells arise directly from the abnormal differentiation and accumulation of oncogenic mutations on neurostem cells or neuroprogenitor cells and their glial progenitor progeny. Germinal centers containing adult neurostem cells have been considered a source of human gliomas because of the clinical observations that glioma tend to be either periventricular or contiguous with the subventricular zone. Strong evidence in animal models supports the idea that regions of the brain with a high degree of cellular proliferation are consequently more susceptible to malignant transformation by chemical or viral exposure. Lee and collaborators recently demonstrated that radiologically and pathological normal subventricular zone cells obtained from patients with IDH wild-type glioblastomas contained low level of glioblastoma drivers mutation that were found to be at high level in their corresponding tumors. Using a mouse model, the authors also demonstrated that astrocyte-like neurostem cells migrate from the subventricular zone to reach other brain regions to give rise to a tumor. Primary brain tumors contain cells of various origins. It has previously been established that brain tumors locally release multiple growth factors and chemoattractants, and they have been implicated to form defined angiogenic niches within the tumor stroma. Additionally, physiologic adult neurostem cells have an uncanny ability to migrate selectively toward brain tumors. These suggest that the tumor cell of origin may not directly divide or originate from the tumor mass. Rather, this brain tumor initiating cell may work to form tumors by locally attracting adult neurostem cells into a spatial brain tumor stem cell niche. The tumor initiating cells then locally release a horde of growth factors into the surrounding area, inducing the previously normal neurostem cells to proliferate in an unregulated manner. This unregulated growth subsequently leads to the formation of the tumor mass. Neurostem cells and brain tumor stem cells share many of the phenotypic properties essential to their maintenance and proliferation, making it plausible that these two sets of cells share similar intracellular molecular pathways. The best characterized pathways that are implicated in the formation of brain tumors are the sonic hedgehog and notch pathways. These biochemical pathways are implicated to be important in the pathogenesis of medulloblastomas in particular, owing to the fact that they play a role in maintaining a small subset of brain tumor stem cells in a non-differentiated state and control proliferation. Within the notch pathway, it has been demonstrated that the co-transcription factors FOXG1 and TLE, normally expressed in neural precursors during development, promoting self-renewal and proliferation, are also expressed in undifferentiated astroglial cells in glioblastoma and in brain tumor stem cells cultures. FOXG1 knockdown induces a downregulation of neurostem and progenitor cells markers and proliferation markers, promoting cell senescence and differentiation and reducing brain tumor stem cells-derived tumor growth in mice xenografts. The same effects can be obtained by TLE knockdown. This suggests that such potent gene transcription regulators may be targeted in the future to reduce the tumorigenic potential of brain tumor stem cells in glioblastoma patients. The brain tumor stem cells hypothesis predicts that even if a tumor is reduced in size by surgical or chemical means, if the stem cell regenerating that tumor is spread, the tumor will regrow and the patient will relapse. The failure of current chemotherapy and radiotherapy in some patients may reflect the resistance pattern of that particular brain tumor stem cell populations to standard treatments. Brain tumor stem cells are emerging as an important new target for the treatment of human brain tumors. Diminishing the population of tumorigenic brain tumor stem cells rather than the non-proliferative and transiently proliferative constituents of the tumor stroma can theoretically result in enhanced survival. Identifying specific intracellular pathways that are used by the brain tumor stem cells for growth and proliferation may lead to the identification of chemical agents that can block these pathways and destroy brain tumor stem cells. Alternatively, inducing the differentiation of brain tumor stem cells into more benign, non-tumor initiating astrocytes has been suggested as a therapeutic strategy. Piccirillo and associates have shown that bone morphogenic protein can reduce the number of CD133 positive cells, blocking tumor growth and resulting in enhanced survival of mice by nearly 80%. Recent evidence also suggested that the vasculature of brain tumors creates special niches where brain tumor stem cells are encouraged to flourish and perpetuate. Brain tumor stem cells niche may be critical to the maintenance of malignancies, and that a disruption of this critical niche may serve as a potential therapeutic option.